Welcome back. We've just finished the first chapter on cells, and now it's time to tackle some questions from your recent past papers. Before we dive in, pause the video, download the practice questions from the link in the description, and take a moment to go through them on your own. Try solving them or at least recall the answers. Once you've done that, come back and we'll work through them step by step together. Remember, practicing past papers is a great way to reinforce what you've learned, and always check the official marking scheme to make sure your answers are on point. Please note that the reference years, variants, and papers are listed above each question, and we will begin with the most recent years available. Alright, let's begin. Here's a photomicrograph showing part of a capillary network in a woman. In the image, we see a structure labeled X, which is a red blood cell. It's clearly marked for us, so no confusion there. Now, we're given some key information. The diameter of the red blood cell, the one labeled X, is 10 micrometers in the image, and the magnification of the image is 1250 times. So, here's what we know. The image size of the cell is 10 millimeters. The magnification is 1250 times. Our goal now is to figure out the actual diameter of the red blood cell, basically, how big it really is in its true form. To do this, we'll use a simple formula that is, magnification equals image size divided by actual size of the object. By rearranging this formula to find the actual size, we get actual size of X equals image size divided by magnification. Now let's plug in the values we have. 10 millimeters is the image size while 1250 is the magnification, and it becomes, actual size equals 10 millimeters divided by 1250. When we do the math, we get 0.008 millimeters. But wait, we need to convert that into micrometers. We know that 1 millimeter equals 1000 micrometers. So, 0.008 multiplied by 1000 and we get is 8. And there we have it. The actual diameter of the red blood cell or cell, X, is 8 micrometers. Remember to mention the correct unit if it's not already included in the question, otherwise, you might lose marks for that mistake. Also, it's not enough to just give the right answer. Be sure to show all the steps clearly, write the correct formula, and don't forget to do the conversion. When you include all these details, you'll earn the full 3 marks. Now, let's move on to the next question. It's about a bacterial cell, which is commonly used in biotechnology. The question shows a diagram of the bacterial cell with parts labeled A, B, C, D, and we have to identify them correctly. Please note that, it's very important to read the question carefully because some students might get confused. For example, the question asks for parts involved in the manufacture of proteins, not just any 2 parts. So, you need to label the specific parts related to protein synthesis. We will go through this step by step. First, Part A is the DNA. Part B is the cell wall, and the outer layer is the capsule. This part C is the cytoplasm. Be very careful as some students may confuse the cytoplasm with the ribosome as it's very close to it, but if we notice it, we can see that the line doesn't touch the ribosome, and it's the cytoplasm. The D is the ribosome, which is located within the cytoplasm, and we can easily observe that the line is touching the grain here. So, now we need to identify which 2 parts are involved in protein manufacturing. And we know that the ribosome and the DNA plays a key role in synthesizing proteins. We will write that letter A is involved in protein production, and its name is the circular DNA, which acts as the genetic material in the bacterial cell. Focusing on the role of DNA, write that DNA holds the genes that provide the instructions for making proteins, that is lipase, which is what the question is asking about. So, always make sure your answer directly connects to the question in terms of whatever information is given in the question. Another key player in protein synthesis is letter D, which we call the ribosome, and the role of the ribosome is crucial. It's responsible for linking amino acids together to build proteins. Remember, the main job of the ribosome is protein synthesis, so when you're answering the question, make sure to highlight how the ribosome contributes to this process by assembling proteins from amino acids. It's an essential part of the whole protein-making process. Next, the question asks, suggest why the bacterium named B. subtilis has a flagellum. And the simple answer is, B. subtilis is a motile bacterium, and its flagellar function as a locomotive organ, enabling the bacterium to move. The next question asks, explain why plasmids are useful in biotechnology. Plasmids are valuable in biotechnology because they can be easily modified or extracted. Once altered, the genes or DNA can be inserted into plasmids, which are then introduced into bacterial cells where they replicate and produce the desired product. Let's move on to the next question. In figure 1.1, we have 2 cells from a plant leaf, observed through a light microscope. You'll notice that the cell wall and the cytoplasm has already been labeled for you. There are also 2 other parts in the image that we need to identify and then complete the table provided. A is the chloroplast, and B is the sap vacuole. Chloroplast plays a key role in photosynthesis, containing a green pigment called chlorophyll. This pigment absorbs light energy and converts it into chemical energy, which is then used to produce glucose. While the sap vacuole acts as a food and nutrient storage center. It maintains the turgor pressure of the plant cell, which provides the strength and support to the plant. State one structure that is not visible in figure 1.1 but would be visible in the cytoplasm of the same 2 cells under an electron microscope. In this case, you could mention any organelle not shown in the light microscope image, such as the mitochondria or another structure. However, since the question specifies an electron microscope, and we know that ribosomes are visible under an electron microscope but not in a light microscope, the most precise answer here would be ribosomes, especially since they aren't shown in the diagram either. Let's move on to the paper 1 questions, which labeled sections indicate the sap vacuole, nucleus, and chloroplast. In this question, parts of the plant cells are labeled as X, Y, and Z, and we need to identify and select the correct option that matches each organelle. We know that X is a sap vacuole, Y is a chloroplast, and Z is a nucleus. Match the labeled parts with the correct letters in the diagram to identify their locations. Let's go through this step by step. X is identified as the sap vacuole. Correct. Now, consider options A and B. C and D are incorrect, so we can rule them out. Next, let's compare A and B. Is Y the nucleus? No, so we can eliminate option A. This leaves us with option B, which correctly identifies X as the sap vacuole, Z as the nucleus, and Y as the chloroplast. Next, a new organism is discovered. It contains DNA in a cellular structure. To which group of organisms could it belong and to which group could it not belong? We will proceed in the same manner by examining each option individually. Follow the instructions that indicate the presence of DNA in the cellular structure. From this, we can infer that there is no nucleus, and it is a cell. We will eliminate options C and D because they don't fit. The options are fungi and viruses, which are not compatible with the given description. Fungi are eukaryotic organisms, meaning they have a nucleus enclosed within a nuclear membrane. On the other hand, viruses are not cellular organisms, they lack a nucleus and other cellular structures. Additionally, the information provided suggests that this characteristic applies to prokaryotic cells, such as bacteria, which further rules out fungi and viruses. Between options A and B, option A is incorrect because fungi have a cellular structure and cannot be placed under the could not belong to section. Therefore, option B is the correct choice, as the description aligns with bacteria, while viruses are not cellular structures. Which type of cell has a cellulose cell wall? We can eliminate option A, animal cells, because they do not have cell walls. Option B and C are also incorrect, as bacteria and fungi have distinct types of cell walls. Bacteria possess cell walls made of peptidoglycan. Fungi have cell walls composed of chitin. Therefore, the correct answer is D. A plant cell wall is primarily made of cellulose, a complex carbohydrate that provides structural support to the cell. Next, some young plants were put into the soil and grew well for a few weeks. They then began to show signs of disease. Samples of the diseased leaves were examined using a microscope. Which observations of the organism causing the disease show that it could be a fungus? This question relates to fungal cells, which are not covered in Chapter 1. However, we will be studying fungi in upcoming lectures, so I have included it here to demonstrate how exam questions can sometimes link concepts from different chapters, leading to potential confusion. But don't worry, if you have studied all the concepts thoroughly, you'll be prepared. Through this question, we'll gain a brief insight into fungal cells. Look at the characteristics listed in the table. I'll clarify for you whether they belong to fungal cells or not, since you haven't studied this topic yet. Fungi are eukaryotic organisms, meaning their cells have a nucleus enclosed by a membrane. They also possess cell walls but lack chloroplasts. Fungi typically have long, thread-like structures called hyphae that help them absorb nutrients. Therefore, we can eliminate option D because it mentions the absence of long, thread-like structures called hyphae. Option C is also incorrect, as it states the organism lacks cell walls, but fungi do have cell walls made of chitin. Option B is wrong as well, since fungi do not contain chloroplasts. Hence, option A is the correct answer, as the presence of long, thread-like structures, the absence of chloroplasts, the presence of cell walls, and a membrane-bound nucleus all suggest the organism could be a fungus. We will study fungi in more detail in Chapter 2.